Hi, this video is a sort of an introduction to the next chapter of a gene expression analysis, namely single cell expression analysis. I will not show you any scripts here, it's only a talk with a little bit of a history of the subject. It all began with the expression microarrays, which were first made in around 1995, but the massive use started a decade later. Then RNA-seq gradually appeared first under the name next-generation sequencing, and lots of stuff now we do with the RNA-seq, we call it bulk, because later we witnessed appearance of a single-cell RNA-seq, which is another technique, but based on the same principle as RNA-seq, qua sequencing. This linear graph is rather misleading. You might think one technique replaced another and the old one disappeared completely. No, it is not. They still coexist and they serve different aims and goals. The first two methods are based on the discoveries of the 70s last century. Expression microarrays utilize the principles of Southern blot, and the sequencing machines are based on the DNA sequencing by synthesis. Whenever I ask students who are those chops, they usually say, I don't know. The guy on the left is a Fred Southern, and on the right is a Frederick Sanger. Southern invented Southern hybridization, which was a pretty messy method of transferring DNA to the membrane and developing an image by hybridization with labeled oligos. It was a lot of paper wasted and buffers. And Sanger invented sequencing by synthesis with a single-stranded DNA and DNA polymerase. We don't see these gels anymore, but the sequencing by synthesis still exists and operates inside the Illumina machine. So where single-cell RNA-seq comes from, officially it started in 2009 by publication of Tang and co-authors. This paper was not an immediate success. It was a little bit too early, and the claims by the authors were slightly, let's say, puzzling. They compared the technique with the microarrays and claimed that it was not any worse but probably even better, whereas the more proper comparison would be with the bulk RNA-seq, but at that time, 2009, the RNA-seq was also at the very beginning of the massive use. Surely some scientists tried to improve the proposed method using regular tools available in the lab, but as it was demonstrated many times, science makes big steps only when supported by technology, when talented engineers invent some genius machines. And indeed it happened in 10x Genomics when they introduced a Chromium machine. Then the whole business really started making shape and volume. Now we have more names on the market like Takara and others, and the idea of partitioning cells by mixing oil and water in a microfluidics environment probably won the attention of the researchers and engineers. In such machines, cells are encapsulated in water droplets together with barcoded sequencing primers. So then for every different cell we had a barcode which would allow later to demultiplex a regular, otherwise pretty regular bulk RNA sequence data, and then perform the single cell analysis. As usual, there was a question, how do we do the sequencing, and now single cell can do a full length per cell, no problem with this. And the next logical question is, okay, we have something new, how to use it? Every technique has its own niche, its own application. So whenever you ask yourself, what would I choose for my next sequencing, you might think about sequencing costs or some other reason listed here. The real thing is what is your question. Depending on what is your question, you make a proper choice. Microarrays are basically thought to be used for routine analysis of known genes, and currently you can request special designs for special uses like medical diagnostics. RNAseq can do pretty much the same, only it grabs everything what is expressed in your cells, and you can do some discoveries sometimes of some strange kind. And single cell is less about gene regulation and transcript isoforms or anything. It's about cells. That's a big difference. And as a follow-up for this, single cell is more focused on a cell diversity. Consequently, you're not supposed to work with a highly pure and homogeneous population of cells. It is necessary for bulk RNA-seq, but absolutely counterintuitive for a single cell. And then you interpret this data in a direction of mutual relations between different cell populations, maybe finding differentiation pathways between them or predict cell-cell interactions, and not so much about gene expressions. You have to be careful. The size of a data is expanding quite dramatically. Very likely you will need more powerful computer than before. You will discover new data formats in this corner. The protocols for data analysis are still under construction and some steps are not very clear. And you will discover more differences. Whereas in a regular bulk RNA-seq, dimensionality reduction plots did not play any significant role. Rather, model design and contrast were in the center of analysis. In a single cell studies, the dimensionality reduction plots are central, and all those clouds on those plots are of a major interest. What people do not realize, sometimes maybe young students, that by manipulating with the settings, you can manipulate with the clouds, and therefore some phantom clouds can appear which have little to do with reality, and you have to be careful declaring something which might not exist at all. That's all what you have to know before you make your right decision. And I will stop here.